Yes.
So, and I think now I'm kind of getting more, I oscillate, right? And now I'm getting more excited about that. And again, one of the drivers back to how do you choose projects, part of it is how excited are the people around you for working on that project. And so that's a big driver. You can have all the ideas you want, but if no one wants to work on them, so what, you know? So we've had a lot of renewed interest in sustainability work. And of course, several years ago, together with Dan Sarah in the chemistry department, we invented the Bionic Leaf, which is the interface of living bacteria with an inorganic electrocatalyst that can be driven by sunlight to split water and make hydrogen. We've gone on to do that, have similar bacteria that can not only use the hydrogen, but they fix CO2 and nitrogen. And those can be used as a very robust nutrient for plants in soil. And so Dan and I co-founded what's turning out to be a wildly successful company called Kula Bio around that concept. But meanwhile, within the lab, we've continued to pursue ideas around engineering those bacteria to potentially, for example, make foods. And I have two amazing former postdocs, Marika Zizak and Shannon Nangle, who have engineered them to make different versions of milk fats. And so this is a totally sustainable green method of using gas fixation to make milk fats. And they just successfully spun out their company, Circe, which I'm really excited about. And then, so the overall theme is enhancing carbon fixation and using carbon via gas fermentation or using hydrogen to make stuff. So that's one of the big themes. And then the other that we sort of stumbled on a few years ago was this idea of cell or organoid or whatever preservation and mimicking. We like to do things that mimic how nature does things. And we were inspired by the lowly tardigrade, which is a water bear, for those of you who don't know. These are amazing organisms that can undergo desiccation and come back to life. And they're the ones that they take to the moon and all that. And it turns out that the mechanism for that survivability is through proteins. And so we have embarked on a project to use those proteins and other derivatives and synthetic versions to preserve cells through desiccation and other kinds of stress.